Thank you for the nice introduction, and thank you for Daniel and Greg organizing this excellent conference. I will see lots of information in a single day. I'm quite tired now. Hopefully, my brain will continue working during my talk. Okay, yeah. So, as an introduction, I'm Hao Cheng. So I'm a associate professor in Department of Animal Science. I apologize. Most of my examples go to animal genetics, but I do have a few examples in plant breeding. But I do think lots of methods we can borrow, across different domains. Usually in my talk, this is always the first slide I show. So this from a paper about 100 years of statistical development in animal breeding and genetics, but generally I would say genetics and breeding. If you look at this kind of 100 history, if you read a little bit, of course this summarized by ChatGPT, okay? If your contribution is ignored, not my fault, okay? ChatGPT summarized it. But if you look at these details, linear mixed model, as we have seen lots of talks about that, and really play a very important role in this, okay? And as I just mentioned, my advisor is Rohan Fernando, and my grand advisor, so kind of like Dan Ginola, I always quote his sentence here. So he said, the science of genome-enabled prediction has reached a reasonable destination, but the voyage will continue. And new data will bring challenges. Okay, so let me think about, okay, in terms of conventional data we have been working on, let's say pedigree data, genotype data, let's say numeric phenotype data. We record this kind of phenotype as numbers. So how about new data? What type of new data? Or let's say at that time, let's say some unconventional data stream we can deal with is also the topic for this section. Something at that time I can come up with, let's say this kind of molecular phenotypes, let's say RNA-seq information, or let's say metabolic data, microbiome, and nowadays, let's say single-cell analysis, let's say single-cell transcriptomics data. Further, as I promised, I will have some plant data here. So let's say image data. This is strawberry data. I will show you some application of our method for strawberry. As well as, let's say, test description of this kind of sample. Let's say this is a description of this strawberry. Okay. Furthermore, I think my collaborator, Dr. Richard Moore, is also here. We work on something about data encryption. How do we use encrypted data in genomic prediction instead of the raw data? Okay, so these are going to be the overview in terms of data I'm going to talk about today. Let's get started with intermediate omics data. We deal with genotype, SNP, and phenotype. But in between, we have this kind of intermediate omics feature, epigenome, transcriptome, metabolic, and all this kind of data set. When we see this data, because we are so familiar with this kind of mixed model, so intuitively, one thing we want to say, okay, how about we just throw in this data into the mixed model, framework, okay, before we have this kind of genotype here on the right-hand side, we have the phenotype Y on the left-hand side, and we throw G-block model. We talk a lot about this one today, like the Bayesian alpha-beta model, okay? So in terms of building the link between genotype and phenotype. On the other side, this is about mixed model, something we cannot ignore. I also saw a few talks today. So let's say nowadays, I saw we also have talk, tomorrow or on Friday about, let's say, this kind of foundation model, let's say for bioinformatics, triggered by this kind of AI, or let's say deep learning, or let's say, just now I heard this kind of large language model for making the breeding program. It's kind of like a AI agent. So with AI, how about the AI agent we can use? But meanwhile, I'd like to say, let's say this is a paper I saw recently published this year. So it's a Nature method about, okay, in terms of gene perturbation prediction, deep learning-based model does not yet, let's say, outperform a simple linear baseline. I would say we are kind of at the stage, linear models are still very, very powerful, but there's the emergence of this kind of, new technology, AI, deep learning, or neural network. Okay, so Given that background, what is the best approach, okay, for us to incorporate this kind of intermediate omic data? If we stay with a linear model, let's say we have this kind of linear mixed model, we have Y, we have the left-hand side and right-hand side. We throw the genotype, phenotype there, but now we have this kind of intermediate omics feature. So where should we put, this intermediate omics feature? So there are different ways I have seen from different papers. Let's say we can feed this kind of intermediate omic feature as additional random effect. Consider this as additional component in addition to genotypes. Let's say we feed this kind of large amount of intermediate omic feature as additional traits. So do this as a multi-trait analysis, but of course with hundreds of thousands of traits. But in my mind, I think a better way to do it will be to develop a unified framework to integrate this kind of, we got this question, multi-layer of heritable information. In terms of molecular, let's say, why do we choose a site? Okay, maybe we should consider model those as a sequential layer of data following the central dogma. So why don't we feed the intermediate omic feature in the middle between the genotype and the phenotype? So that's how we develop the model NMM, so neural network with mixed models. Now I will briefly talk about it. So NMM is a model we developed to extend mixed model to neural network. So this is the general framework of it. Here we are showing you a 3-layer neural network. But in the middle layer, we are fitting the intermediate omic feature, or let's say molecular phenotypes into it. If you think about the input layer and the middle layer, what we have is a single trait mixed model. Okay, and let's say this for the gene expression of one gene. So between these, we fit a single trait mixed model. Between the middle layer and the output layer, we allow this kind of activation function in different neural network, let's say tangent, sigmoid, this kind of nonlinear activation function. NMM, our model can handle missing omic data. When we deal with omic data, for sure we'll have this kind of missing problem. Either because we have limited resources, we will only be able for some individuals. So, so we have this one, so it will be able to deal with different patterns. Let's say all omics observed on all individuals. Let's say all omics observed on a subset of the individuals. Let's say random missing. Let's say no omic data. But anyway, this one is dealt with by through a Bayesian way, through MCMC sampling. Here's a simulation result we show here. The x-axis here is the missing rate of omic data, jumping from 0 missing to 100 missing, and the y-axis is the prediction accuracy. What you can see here, the GBLUP method using genotype only serves as a baseline. This accuracy if you use genotype only from our simulation data. But you can see NMM using omic data generally provide higher prediction accuracy in this simulation, and the lower the missing rate of the omic data, so the higher the prediction accuracy. It's kind of a general pattern here. Anyway, these are simulation data. Some other features will be, let's say NMM is equivalent to a mixed model. If we assume the middle layer there's no data, and we put a linear activation function here. So everything here are linear, and then we are going to see same result as the mixed model. NMM allow different priors because we've been working with mixed models, so it allows, let's say, base C prior, mixer prior, it allows GBLUP prior, those kind of different prior. Okay, depends on what the true genetic architecture is, we may see different benefit. We allow different activation function. Again, we never know the truth, but we might be able to try different activation function to see the performance. So here I want to show you a few real data applications of NMM. The first one I want to show is one of our projects. It's supported by USDA. We work with a pig breeding company called PIC. For this one, we work with about 2,000 peaks, 40K marker panel, and we collect metabolic data from this peak population twice, one at 10 weeks and one at 20 weeks, okay? And then finally, we get this kind of about 100, molecular metabolic profiles across these 2 time points, and then they provide us a few feed efficiency traits related to this project. NMM will provide you the heritability estimation from a Bayesian way. Generally, I would say, can you see the number clearly? Looks like. So generally, I would say the heritability, the green one is heritability distribution for 20-week metabolic profile. The red one are the 10-week, okay, heritability for 10-week metabolic profile. So it's generally between 0.1 to 0.3, and the 10-week one is a little bit higher than the 20-week one. The figure B shows you a little bit detailed information in terms of making a scatter plot, but basically telling the same information. So generally, these kind of metabolic profile are heritable. Okay, so as long as these are heritable, we are willing to feed this one to our NMM model. This is the figure in terms of, prediction accuracy. Let me see whether you can read this. So I just briefly walk you through this one. Each panel represents one trait. The y-axis, I'm showing you the accuracy difference. Basically, the baseline model is a G-block model. Okay, so show you how much better this model is compared to G-block. So the blue ones are the method using metabolic data only, so no genotype. Generally, you will see how it performed worse than the baseline model. But the yellow bars are the method using NMM, so fitting both metabolic data and genotype data. I will say generally, we have 3 here. So the first one is like genotype plus 10-week metabolic data. The second one, genotype plus 20-week metabolic data. The last one is genotype plus 10-week and 20-week metabolic data. Basically, it really depends on which metabolic data we're using. Generally, I will say the last one, so when feed all metabolic data and all genotype into a model, it reaches the highest prediction accuracy. But it also depends. Let's say for this trait, okay, average daily feed intake, 20-week and 10-week and 20-week together plus genotype provide almost similar prediction accuracy. But this one it varies. Okay, it's really case by case the result. If you have a look at this one, average daily gain. So by combining both using genotype plus 10-week metabolic data or using genotype plus 20-week metabolic data, both give you a significantly higher accuracy, but combine the 2 time point metabolic data together, you are going to see a little bit higher accuracy. I would just say when you incorporate intermediate omic data into the picture, usually you need to study this one case by case, which molecule you are using, or let's say even which trait you are studying, and of course the time point. This is the one I saw recently, published in GSE. For this one, they use NMM for sheep microbiome. They have about 12,000 SNPs, a large number of microbial features, let's say 200,000 microbial features, and about 1,000 animals. So for this one, they deal with this kind of methane or methane ratio or CO2 trait. Generally, what I can see here, maybe I can read this one a little bit for you. Oh, this one is clear. So the first one is genotype only result. The second one is genotype plus omic data, but fitting in a linear model way. So basically fitting omic data as additional random effect, okay, to the right of the mixed model equation. And then because they have a large number of microbial features, so they do PCA, okay, for the microbial features. And it looks like when the number of PC is 88, and these are fitting to an MM, okay. When the number of features 88, magic number. So maybe reach to some variance level. So the accuracy is generally higher than others. Okay, than the baseline model, or better than the G plus M, but mixed effect model. But only thing is like when they have more PC, the accuracy really go down. One reason might be because of the later the fewer variance explained, maybe the noise signal ratio becomes very bad, so you won't be able to bring lots of benefit. Anyway, this is one example. I don't read this paper carefully, but roughly this is the one they have been working on. Another one, because this one uses microbiome and looks like they do PC, so I talked with somebody I know from University of Nebraska Lincoln for personal communication. This is the result she showed me. So they are dealing with similar question, but in cattle. So that's for cattle microbiome. When they deal with PC, fitting PC into for the intermediate omic data into NMM, looks like they don't always get good result, but actually, finally, their solution is even much easier. So they just find the top, okay, microbiome features. So for these are about top 100, top 300, but generally you can see so with just fitting the top, let's say microbiome features without doing a PCA, they reach a much higher prediction accuracy and also significant. Okay, this is another example. I don't have time to jump to all the detail, but something else we are working on. This is for prediction, but NMM is also quite useful for gene trait association, let's say TWAS, transcriptome associated study, also fine mapping. So one thing we have been working on is like we are incorporating this kind of public information called farm GTAG. They have this kind of gene expression information from livestock and incorporate that one into TWAS analysis. NMM deal with this one in a nice way. In a sense, we are going to fit all genes together, okay, here, and plus a direct effect from the SNP to the trait. So usually nowadays when people do TWAS, they deal with one gene by one gene. So it's a single gene analysis. We have seen some results. We have the power is much higher. Another one, this one is dealing with human data, but this one is really more about, I heard that Pleiotropi talk. This one is about quantifying annotation using this kind of single cell transcriptome annotation for cell type to further dissect Pleiotropi into shared polygenicity and and this kind of coherability. Okay, these are some examples. So this is everything I want to talk about molecular phenotypes. As what I promised, now I will start talking about some plants. Okay, so the reason why we come up with this project because we have a strawberry breeder on campus at UC Davis, and he has this kind of data set. We are wondering whether we can do something together with this breeder. So then that's actually come to the picture. So we have this kind of image. We may have this kind of test description from the breeder talking about, this sample. So the question becomes, okay, can we use NMM for images and text? Okay. Instead of just putting numeric phenotype there, let's say body weight or something, milk yield or something like that, or plant yield, can I just put the image directly there? So I'll be able to feed this one into a nice framework. Finally, based on the genotype, I can predict you the image, or let's say predict you the text description. By talking with the breeder, we feel like there's lots of benefit. For example, it will be able to capture complex and subjective traits, let's say shape, or let's say color, or let's say disease symptom, animal, let's say we care about, let's say animal behavior, beyond, let's say numeric numbers. And I know for breeder, when they convert this kind of image into a single number, it may have this kind of information loss there, or maybe it introduces a human bias. You ask the same person to evaluate, different person to evaluate the same image, they may give you a different, subjective rating, okay, but because you are converting a quite information-rich image to a single number. Something I also like is about the improvement of interpretability. So because with this one, we predicted we will be able to show you a predicted image, or let's say predicted, sentence description. At least for me, I think who are not familiar with strawberry, it's more direct, okay, more straightforward. Okay, so one example is like this. So at least when I work with him, he showed me these numbers. I said, oh, they're numbers, but I really don't know what are these numbers, okay? Let's say length-width ratio, RGB, the color channel, shape index, but these are for two strawberry, okay? But it's really hard for me to tell which one is which one, or which one is better. But when he showed me this, I think, okay, even as somebody who just eat strawberry, okay, I can easily tell by reading, yes, but even from the image, I can easily tell the first one. I guess this is the one I want to go for if I'm a breeder. Then we get started. So we get started with, typically how people deal with this question. So they got image, but they use some computer engineer package, or let's say they do something to subtract the information from the image, or let's subtract the information from the text description, and then they throw these numbers, okay, to mixed model, and you will be able to predict these numbers. That's a mixed model way. But what we want to do is like we really want to, okay, we throw the image and text into the algorithm. And finally, for my testing population, I provide you my genome tab. You are going to send me show me the predicted image and predicted text description. That's kind of the idea behind it. We got started with, predicting the raw image. When I say raw image, let's say we have this kind of, for sure it won't work. Let's say we have this image, but we just get each pixel, okay? And we just treat each pixel as a trait, and link that pixel to the genome tab. Turns out this is what we got, okay? It's like just noise, nothing, okay? Yeah. So, for the text, same thing. We know that's one word, but we just want to show it, okay? It's like we just get the character, okay? And then we throw the character into the model, and we predict it. Of course, the prediction is also doesn't make sense at all. For this one, then, so then we start this project. We term it kind of end-to-end genome prediction, but really the only idea is really about using the NMM framework, but do direct prediction of image and text directly from a SNP marker. Okay, so whether we can feed this one here. Then we borrow something from the AI domain. We are trying to see whether we can make this kind of multimodal phenotypes low dimensional and heritable. Okay, for this one we are using a tool called, we didn't code this one by ourselves, we're using some existing tool to do it, so called autoencoder. So for this kind of autoencoder, in short, it's kind of nonlinear PCA. So basically you will be able to reduce this, image into a few embeddings, but with a decoder, you can convert your embedding back to the image. Okay, as long as the performance of autoencoder the accuracy is high, you'll be very happy to do it. This is for the image part. For the text part, we are using this morning mentioned that we are using a pre-trained large language model and ChatGPT to do that. Yeah, so once we finish training that one, the only thing I need to do, okay, is like to incorporate this one to NMM is really, I just need to copy or let's integrate that part or that weight into my NMM, and of course getting the embedding there for further training. Okay, so then we start our analysis. I would say this is really a small data set. If you have a bigger data set, you are wanting to try this one, and please contact me. So it's really we have only 500 strawberry, to be honest, and then but for each strawberry we have about 20 images. Okay, and then the internal number of images is a decent number, but in terms of genotype, we only have, let's say, 500 different genotypes. We get of course, from the image we can get it, but at that time we don't have we're generating it, but at that time we don't have the text description. So we just ask ChatGPT to generate the text description from the image. Okay, so basically now we have a text description data set, we have an image data set. Oh, we have 50K marker for the strawberry. For our method, essentially, we really don't need to get this trait, let's say length-width ratio, RGB, or whatever. So because we predict the image and text description directly, but just to benchmark it, okay, how good it is. Okay, I can anyway provide you a predicted image, but it's really hard for us to evaluate how good it is sometimes as human being. So basically, what we did is like we define a field trait, the breeder define a field trait. So then for the image or text, we get those traits by measuring it. And then we run a conventional G-blob or base C model there. And we also do the same thing. Again, we are just training our model using the image and text. But for our prediction, as for our predicted image, we can get the same value. We can get the length-width ratio from our predicted image. Then we want to see, okay, if we are doing a good job, hopefully that value is closely, can reach, similar accuracy as the conventional method. This one is about accuracy for the embedding step. Just remind you, embedding step doesn't involve the genotype. So for this one, it's really about, we do this kind of auto-encoder, how good it is, how good we can represent the image through a low-dimensional embedding. So it reached about, I would say that we need more work, but it reached about 0.7, 0.8 accuracy. And this is generally the prediction result we got. Again, the red line is a baseline. So basically, the accuracy you got for each trait from the conventional, let's say G-blob or base C model, and the plot here shows you across different, validation, the accuracy or the accuracy difference from our end-to-end method. I would say generally for some trait we do see equivalent or a little bit higher prediction accuracy when we compare our one to the conventional method, but the benchmark we are using is really in favor of the conventional one because we are getting numbers from those images to make the comparison fair. I would say for some situations the accuracy is a little bit lower, but I think for text the performance is a little bit worse. But generally, I think it's acceptable. At least serve as a prototype by feeding this image and text into the prediction. I think as a prototype, I'm quite happy with this result. We have this kind of, we can show you because behind it we have everything, and this kind of showing you behind the MMM with image what's happening there. So show you finally the predicted image, or this is the original image. These are for the testing data set. Okay. Am I running out of time? Okay. I have 10 more minutes? Okay, sure. Yeah, so, yeah. So this is about some example here. So the last project I want to mention here because Dr. Richard Moore is also here. Dr. Richard Moore and I we have been working on this method for data encryption. So we can use encrypt data for genomic prediction and GWAS. It's another type of, I call it unconventional data stream. So actually this project is one part of a USDA project I'm currently leading. It's about G2P, data integration, sharing, and collaboration. I think I will briefly talk about it. We also have a poster here. So we have way more details in that poster. You can go to that poster during the post session to get more information about this project. Generally about our data sharing project is a collaborative work. So we are trying to build platforms and methods for data sharing using both public data and confidential data. We also work with a few stakeholder advisory board about the need and the potential for us to make it work. Okay, so data sharing. Maybe I can skip a few slides if I'm running out of time. So as we know, data sharing is really important. What I'm showing here is like no matter if you're going to publish a paper, they have some requirement to ask you to make the data publicly available. Or let's say getting grant. I know the grant situation now in US is disaster, but in terms of getting grant, you also have this kind of requirement. Okay, so you need to make your data available as soon as possible because those are supported by the federal grant. Okay, thanks. And we know the benefit of data sharing, how it can validate of results, reuse of data, joint analysis, let's say for collaboration. A good example is GWAS. So by having, a larger and larger sample size, there are more, GWAS loci we have been discovering. This for human genomics. On the animal side, for dairy cattle breeding, so we have the CDCB in US. This is also a joint effort. So farmers share data with CDCB, then CDCB will be able to do joint genetic evaluation to make it more powerful. So we can reach, I think somebody mentioned that for dairy cattle, we have this kind of big data set, so we'll be able to reach a very large data set. And there are restrictions. So confidential content, less private content, commercial sensitive content. So that's the reason why we work on this one. Currently, we are building this website. It will be released early 2026. We have a few features there. For example, we are making a web app to host, public data set we can find online. Now we have about more than 100 public data set metadata there, so you'll be able to download this data find based on species or sample size. You can search it, and then you'll be able to download the data set. And we also have some associated data analysis code there. For example, we also we start talking with companies about building this kind of commercial data access request portal. So researchers can submit a proposal through us to the company to request that data. And we are talking about some details with companies about, let's say, how about we can build a tiered data access model. Let's say we may have different risk level. Okay, maybe for your raw data, the risk level is so high. We need a very strict, MTA. But how about we could encrypt the data? So how about, I'm not saying you need to make your encrypted data public, but how about with the encrypted data, you can make the MTA, let's say data transfer agreement, much easier, much straightforward. So that's kind of the reason why we really want to work on this kind of encrypted data sharing solution. Like I mentioned, originally this method that I've been working on was developed by Dr. Richard Moore, HEGP, homomorphic encryption for genotype and phenotypes. And later we further extend this one, develop this one to make it work for genomic prediction or GWAS and most models we have been using for in breeding. So in short, really this one really allows data sharing and joint analysis, but protecting the confidential information. I do have a
Nowadays, phenomics and genomics data are highly used in genome to phenome analyses. When publishing scientific articles, many journals are also interested in and may require the publication of the corresponding data, consisting of genotype and phenotype information, as this allows for reproducibility. However, companies and institutions may not want to publish their raw data for privacy reasons. A similar situation arises in the case of joint analyses, where several organizations pool their data together. These organizations may also not want to share their unprotected data with others. One solution to these privacy concerns is implementing an encryption scheme that allows for outcomes identical to those that would be obtained without the encryption, such as estimated marker effects, while protecting the data, such as keeping it encrypted during the entire process. A common method that has been used before is de-identification, where identifying information, such as names, is removed. This method is useful in making it more difficult to identify individuals in the dataset. But a downside of methods like this is that after de-identification, one can still infer information about individuals, for example, relatedness with other individuals in the data set, which we would like to avoid in order to keep the data secure. A new method that we are developing, homomorphic encryption for genotypes and phenotypes, abbreviated HEGP, allows you to obtain the same results from the encrypted data as those obtained from the unencrypted data in the form of estimated marker effects. In addition, individual information can't be successfully inferred from the data encrypted with HEGP, especially as the data remains encrypted throughout the entire process. To demonstrate how HEGP works, we'll take an example of one trait controlled by one SNP, shown here on the right and the left, respectively. First, we'll center our data and plot it on a scatter plot and fit a linear regression, where the slope of this line represents the marker effect for this particular SNP. After implementing HEGP, this is how our data changes and how it looks after being plotted, represented here as triangles. These data points look quite different, but after fitting a linear regression on the encrypted data, we can compare the regression lines we get from our data before and after encryption. As you can see, these two lines overlap, indicating that the slopes are the same. Another way to put this is that the estimated marker effects we obtain from the unencrypted data and the encrypted data are identical. Although this example only features one SNP, we have previously demonstrated that HEGP can be successfully implemented for genomic analyses that use information from many SNPs, for example, genomic prediction.
Okay. Yeah, again, AI-generated audio. Yeah. Because many of you here are familiar with this kind of mixed model, so actually the method is really quite simple and straightforward. I guess it's also because it's simple and straightforward, so then we'll be able to implement it easily. So it has lots of potential. The idea is really we have the genotype and phenotype here, but we can generate some random orthogonal matrix, basically P. So P transpose P is identity. So then what we do is really we just pre-multiply the genotype X and Y, genotype matrix and phenotype by P. Okay. If you think about it now, you guys know this mixed model. Now you're solving normal equation. Your left-hand side and right-hand side will stay same. So basically, when you solve your market effect, you will get the same answer. Yeah. When you deal with different cases, they become more complicated. But in short, this is kind of simple explanation. Yeah, and we don't need to. We just share the encrypt data, but with the key, we don't need to share the key. Okay. So with simulation, we show that. For the estimated breeding value across different genetic architecture, we get accuracy close to 0.99, so close to 1. And we show with real data. These are real data. This is kind of real case. So pig gene, at that time, they have 6 companies there, and they want to do a joint analysis for natural disease challenge model, but they don't want to share the data with each other. What they can do is, like, if that's the case, they can encrypt their data, and they can feel comfortable to share with each other, okay, if they want to do a joint analysis. So for this one, it's kind of like we did a joint analysis for these kind of 6 companies by combining their data together. So the plot I show you here is like on the left, because this is mainly for GWAS, on the left is the GWAS using unencrypted data. On the right is the GWAS using encrypted data, but roughly from this kind of Manhattan plot, the results stay almost identical. Okay, this is kind of for the encryption one. As mentioned, we have the poster. If you want to know more, please come to the poster session. So in total, I talk some of these maybe you don't consider this as unconventional anymore, but at least in addition to genotype and phenotype, we talk about molecular phenotypes, we talk about text, we talk about image, and this kind of encrypted data. So I think that's everything I want to talk about. Feel free to ask any questions. Thank you.